I mentioned earlier in this lecture that one of the glial transmitters that's released from astrocytes is D-serine. Turns out that D-serine plays a very important role in the activation of NMDA receptors. So you know that NMDA receptors are activated when glutamate binds to the glutamate binding sites on the receptor, but in addition there is magnesium block of the pore of the NMDA receptor, and the cell has to be depolarized in order to remove the magnesium block before the receptor pore can open, allowing sodium and calcium into the cell. Well, it turns out that a third event also has to happen before the NMDA receptor can open, and that is that a coagonist has to bind to a coagonist site on the NMDA receptor. For many years it was thought that glycine was the endogenous coagonist that had to bind to the coagonist site. But it has been discovered in recent years that D-serine is most likely the coagonist that is the endogenous coagonist, at least in most parts of the brain, that is needed to activate the NMDA receptor. And D-serine appears to be the endogenous coagonist because it actually has a higher affinity for the binding site, which was previously called the glycine binding site. D-serine has a higher affinity than does glycine itself. So we see that D-serine has to be present at the NMDA receptor in order for the NMDA receptor to open. So the question arises as to what is the source of this D-serine. Well, it turns out that if one does immunocytochemistry, D-serine appears to be localized primarily to glial cells, to astrocytes, and also the enzyme that is necessary for synthesizing D-serine, serine racemase, is also present primarily in astrocytes. And in addition, when glial cells are stimulated by glutamate, this can lead to the release of D-serine. So it appears that the major source, if not the only source, of D-serine in the nervous system is from astrocytes, and this in turn implies that astrocytes and the release of D-serine are necessary for NMDA receptors to operate properly. Now, I should mention that the exclusive source of D-serine from astrocytes has been challenged in recent years, and there is some evidence that neurons can synthesize and release D-serine as well as astrocytes. So this is still somewhat controversial, but it is generally believed that astrocytes are a major source of D-serine. The experiment illustrated in this slide shows that D-serine can potentiate NMDA transmission. These are voltage clamp recordings from retinal ganglion cells, and in this experiment the retina was stimulated by light, leading to a inward current in the retinal ganglion cell, which would depolarize the cell. Under control conditions, we record a modest inward current in the cell. If D-serine is added to the retinal preparation, this inward current becomes much larger, and then if D-serine is washed out, the response gets smaller again. So this demonstrates that the D-serine site on the NMDA receptor is not normally saturated, but if one adds additional D-serine to the preparation, then this activates this coagonist site, leading to greater NMDA synaptic transmission. So again, this demonstrates that D-serine plays an important role in modulating and controlling the amplitude of NMDA synaptic transmission. Scientists have taken advantage of a unique response of the hypothalamus to demonstrate the importance of D-serine release from astrocytes. in certain nuclei of the hypothalamus, including the supraoptic nucleus and the paraventricular nucleus, glial cell processes surround neurons as they do at other tripartite synapses. However, during parturition, that is when female animals give birth, and during lactation, these astrocytic processes are withdrawn from the synapse, from the tripartite synapse. Now, if D-serine is released from astrocyte processes at the synapse, and if this D-serine is important for NMDA receptor transmission, then during parturition and lactation, with the withdrawal of astrocytic processes, NMDA receptor transmission should be reduced. And in fact, this is exactly what is seen. During parturition and lactation, there is a decrease in NMDA receptor transmission, and this decrease is due to a reduction of D-serine at the synapse. In addition, LTP is reduced during parturition and lactation, and this is also due to a reduction in D-serine and a reduction in NMDA receptor transmission. In the next several slides, I will illustrate experiments that demonstrate the importance of D-serine at these hypothalamic synapses. What's shown in the illustrations at the bottom of this slide is the localization of D-serine to astrocytes in the hypothalamus. What's shown on the right in figure D2 is GFAP labeling, which highlights the astrocytes in the hypothalamus. GFAP is an intermediate filament found exclusively in astrocytes, and the red label shows the distribution of astrocytes in the hypothalamic nucleus. What's shown on the left in figure D1 is the distribution of D-serine in the same sections with an antibody against D-serine, and as you can see, D-serine is localized to the very same cells that is labeled by GFAP, demonstrating that D-serine is contained in astrocytes in these nuclei. This series of records shows the importance of D-serine release from astrocytes for NMDA transmission in the hypothalamus. If we look at the upper left here and these records, these are voltage clamp records that show both an AMPA component of synaptic transmission and an NMDA component of synaptic transmission. The details of how these recorded are not important. For NMDA, the neuron was depolarized, but the important thing is that one can isolate and measure the separate components, the AMPA and NMDA components of synaptic transmission. So this is the two components, the magnitude of the two components in a virgin female animal. In the next record, in the middle record on the top, we see the same records of synaptic transmission in a preparation where D-amino acid oxidase, or DAAO, is added. D-amino acid oxidase breaks down D-serine so that D-serine levels will be reduced. And under the conditions where D-serine is reduced, we see that the AMPA synaptic current remains the same, but the NMDA synaptic current is much smaller. However, if we then add D-serine back to the preparation, we can increase the magnitude of the NMDA component of synaptic transmission. So this experiment shows that D-serine, endogenous D-serine in the preparation, is necessary for proper NMDA transmission. Then in the right-hand diagram figure on top, we see records of synaptic transmission in a lactating animal. Now remember, in a lactating animal, the astrocytic processes are withdrawn from the neuronal synapses so that D-serine will not be present. And here we see the same type of change where the AMPA component of synaptic transmission is large, but the NMDA component is very small. Let's go down to the bottom records. The bottom left record is again from a virgin animal. Here's the AMPA component large, the NMDA component is large, and if we add D-serine, we see just a modest increase in the amplitude of the NMDA component. Again, on the right, we see records from a lactating animal where the AMPA component is large and the NMDA component is small. This is the same type of record that was seen up here in the upper right. So, our hypothesis is that the NMDA component is small because the astrocytic processes have been withdrawn. Thus, D-serine is not present at the synapse. So, if we added D-serine back to the preparation, we should be able to enhance the NMDA component, and that, in fact, is what is seen here. In the lactating animal, if D-serine is added, we have a large increase and a healthy NMDA component of synaptic transmission. We know that NMDA receptors play a very important role in synaptic plasticity and in particular in generating LTP, long-term potentiation. So we might expect that synaptic plasticity in the hypothalamus might be altered during lactation when the astrocytic processes are withdrawn and D-serine at the synapse is reduced. And in fact, this is exactly what we see. In the left-hand panel, we see an experiment that shows induction of LTP in a virgin female animal where with tetanus we have an increase in synaptic transmission that is maintained for, in this case, 30 minutes. So this is the standard type of LTP that is seen, for instance, in the hippocampus. In the lactating animal, in contrast, when we apply the same tetanus, we see not an increase in synaptic transmission as was seen in the virgin animal, but in fact a decrease in synaptic transmission. And this, again, is hypothesized to be due to a withdrawal of the astrocytic processes, therefore a reduction in D-serine levels at the synapse, and to a reduction in NMDA synaptic transmission. And in fact, with low levels of synaptic activation in other parts of the nervous system, we see LTD, long-term depression, instead of long-term potentiation. And this, in fact, is what we see in the hypothalamus as well. So, if this change in synaptic plasticity is indeed due to a decrease in D-serine at the synapses, due to a withdrawal of the astrocytic processes, then we should be able to restore normal LTP by adding D-serine back. And this is exactly what's seen here. This is an experiment done in a lactating animal with D-serine added, and with the added D-serine we see a restoration of normal LTP. So these experiments demonstrate the importance of the release of the glial transmitter D-serine for the normal synaptic plasticity to be seen, and emphasize the importance of glial cells for modulating synaptic plasticity in the nervous system. We've seen that an important function of astrocytes at the tripartite synapse is to modulate synaptic transmission and synaptic plasticity by the release of gliotransmitters. Another important function of astrocytes at the tripartite synapse is the removal of neurotransmitters from the synaptic cleft. And the most important of these transmitters is glutamate, where glial cells, astrocytes, play a primary role in removing glutamate from the synaptic cleft. And this is done by the action of excitatory amino acid transporters on the plasma membrane of astrocytes that transports glutamate from the synaptic cleft into the glial cell, where it's converted to glutamine, and then the glutamine is transported back into the presynaptic terminal of the neuron, where it's converted back to glutamate and the cycle can begin again. This is called the glutamate-glutamine cycle. Now, since the glial transporters are playing an important role in determining the concentration of glutamate in the synaptic cleft, one might expect that these transporters can influence the strength of synaptic transmission. And as we will see in the next slide, this is indeed the case. This figure illustrates an experiment that was done in the retina, recording from retinal ganglion cells. The retina was stimulated by light, and retinal ganglion cells were voltage clamped, and under normal conditions, control here, the onset of light generates an inward current, a depolarizing inward current in the retinal ganglion cell, and then the off of light generates a second inward current. Now, if one adds PDC to the preparation, PDC is a blocker of the glutamate transporter in glial cells, one sees a dramatically enlarged response. So in the presence of this glutamate transport blocker, we see that both at light on and light off, we have a much larger inward current that is generated in the ganglion cells. And this is because with the release of glutamate with light stimulation, the glutamate is released into the synaptic cleft, but it can't be taken up by the glial cell transporters if the blocker is present. Therefore, glutamate remains high in the synaptic cleft for a much longer time, and much larger synaptic potentials and currents are generated. So this experiment demonstrates the importance of the neurotransmitter transporters on glial cells in regulating the strength of synaptic transmission.